Hello everyone. So today my group will be presenting on Bio-compost project number 6. So the members are Tengku Muhammad Daniel, Safri Aiman, and myself is Torili Morin. First of all, we know that biocompost uh, is when plant matter has been decomposed and recycled as a fertilizer. So the general process of biocomposting is done by piling up waste in a field and leave it undisturbed for a certain amount of time. Sometimes takes years or more than that. In this modern day biocomposting, it includes many steps, such as monitoring of the composting, shredding of plant matter, adding sufficient water to maintain moisture level, and also regularly turning mixture to provide aeration. Okay, there are wide range of bacteria that occur naturally in the soils and will present in compost. So there are three types of microbes, that is compost bacteria, actinomyces, and compost fungi. In this project, in this biocompost, we will be focusing on the compost bacteria, which is lactobacillus bacteria. Lactobacillus. Lactobacillus, it can be produced for lactobacillus bacteria from this biocompost can be produced from fermentation of milk. And here I'll explain some of the characteristics. So the characteristic of lactobacillus is that this bacteria is gram-positive bacteria, and it have rod shape and straight. This bacteria is also fermentative, facultative, anaerobic, or microaerophilic organotrophs. If oxygen present in the environment, the bacterium will undergo facultative anaerobic, where it makes adenosine triphosphate (ATP) by aerobic respiration. If oxygen absent, the bacterium will switch into fermentation or anaerobic respiration, and it can also switch into microaerophilic organotroph, especially when the oxygen level in the soil is lower than the atmospheric oxygen. And generally, this lactobacillus is more tolerant of low pH environment. So we can see here the functions. So what are the functions of lactobacillus? Lactobacillus can produce lactic acid and derive energy from fermentation of sugars to lactate via the homofermentative metabolism. So what is homofermentative metabolism? Which it means that bacteria ferment the glucose with lactic acid as its primary byproduct. Next, lactobacillus also help in cycle nutrient in soil, making so plant have enough nutrient supply. And it also help on degrading organic waste. Lastly, it acts as probiotic to the root of plant by allowing slow release of nutrients and and that it will be absorbed by the plants. Cell function like the rhizosphere. Next, I'll pass it to the next presenter.
Alright, for the process, composting is a method that is known for the treatment of solid waste where organic materials are broken down with the help of microorganisms and also by the absence of the oxygen, which is anaerobic, until it is considered to be safely stored, handled and applied to the surroundings. In other words, it can be used to recycle and reuse organic material. Usually, compost undergoes three phases, which are mesophilic phase, which is moderate temperature phase that lasts couple of days, and then thermophilic phase, which is high temperature phase lasting from few days to several months, and the last phase is cooling and maturation phase, which usually involves several months. Usually, compost process involve four main components, which are organic matter, moisture, oxygen, and bacteria or microorganism. Organic matter is should include brown organic matter such as dead leaves, twigs, manure, and organic materials such as lawn clippings, fruit rings, and many more. The brown materials will be source of the carbon, while green materials supply the nitrogen. For the moisture, if it is too dry, it will decompose slowly. So, but if it is too wet, then the pile should be turned and mixed. And the third component is oxygen. It is important for the breakdown of plant material by the microorganism or bacteria. To do so, the pile needs to be turned over every 2 weeks, and so also to provide the aeration to it. And the fourth components is bacteria or the microorganism. It will these things which is microorganism or bacteria, aka bacteria, will break down the materials into useful compost. And then for manual or methodology, the first thing is preparation of effective microorganism or EM. The first step for preparing the effective microorganism is clean rice with water that do not have chlorine, and the wash water is being collected and stored in a container for 3 to 5 days. And secondly is put the wash water, then put the wash water and fresh meat in a clean container with a ratio of 1 to 10 and store for a week. After a week, 2 layers will form, which consist condensed milk and yellowish solution, which is lactoserum, containing the bacteria we need, which is lactobacillus. So filter the solution and collect lactoserum in another container, another clean container. And the molasses, which is some type of sugar, will add into the lactoserum with ratio 1 to 1 and ready to be used to in compost. And then for preparation of the compost, first thing to do is prepare a pile of soil layered with dry leaf containing carbon source and green grass clippings as the nitrogen source, as explained in first part of mine. And then put the EM that had been made before into the pile and leave it for composting process to start. And after the mix had been done, observe and record the change it could occurs during the composting process.
Okay, next I will be continue on planning. So there are 7 steps in planning for the compost. So the first step will be type of compost bin. So we can get variety of compost bin from the garden and house store. So we need to decide which type of compost bin that we should buy. So because of that, we can decide the best will be the one that is neat, can get the animal out of that compost bin, and also the one that can preserve heat. And then the next step will be location of composter. We need to plan where we want to put the compost. So the suitable location will be the one that is flat and then well drained and can get the sunlight. And most important, it is a convenient place or convenient location. And then the next planning will be the alternative layer. So alternative layer is a layer where we put for example like dry leaf. So for between of each green component, we can put a brown component. So the alternative layer is the brown component. So this brown component can be good aeration for the water to flow, so that the compost bin will not be too wet. And then after we have put the alternative layer, we can put waste. So we add more waste. So we can put our kitchen waste, our garden waste, and then after we add waste, we add it until full. And that is the next planning step. We add until the compost bin is full. And then the next step will be maintain. So we need to maintain our compost bin. So for every time we add our waste, we need to flip the compost bin so that the old waste that have been added will be mixed together with the new waste that we add. And then also we need to add more dry leaf or brown component when we see that the compost is too wet, so that will be a good aeration for the compost bin. And lastly will be the harvest. So when do we know when we want to harvest our compost bin? So we can look at the result of the soil. It will be dark, crumble, and it will smell like earth. So that is the seventh step of planning the compost bin. So we pick the type of compost bin, and then we think of the location, and then we pull the alternative layer, and then we add waste until it's full, and then we maintain it, and then we can harvest it. Okay, the next one will be assessing biocompost. So how do we know that it is a good biocompost? So the first step is seed germination test. So we can take the final result of the compost, so the soil, and then we try to put a seed, and then we when the seed grow, or if the seed is not grow, so from that we can also observe if the seed grow or not. And then if the seeds grow, we can measure the length of the root, the length of the shoot, and also how many leaves that grow, and many more. So this is the first step of assessing our biocompost. And then the next one will be the respiratory analysis. So in this, we can use the specific oxygen uptake rate, SOUR, by measuring the rate of oxygen consumption in the liquid state. So we put the soil into water, and then we measure using a respiratory meter how many oxygen are being consumed by the soil or the organism in the soil. So next we will talk about the issue. So the first issue is soggy compost. So it will cause the microorganism in the compost to be lack of oxygen. So to counter it, we can add more dry leaf to give a better aeration. And then the next one will be a smelly compost. So if our compost smell like an ammonia, it's an indicator that the compost is containing too much nitrogen. So we can add more carbon like a leaf. And next, the next one will be too acidic. So usually our compost is naturally slightly acidic. But sometimes if we add too much water, it can upset the balance. So when this happens, the compost will be smelly and usually slow to degrade. So we can add more carbon and nitrogen to counter the acidic. And then the next one will be finished product to coarse. So some materials such as eggshell usually take a longer time to break down. And also we should never stamp or force the material down in the compost to feed more. And lastly will be compile pile won't heat up. So this may cause because the material in the compost is too dry. So we should always keep the compost material moist, and maybe it is low in nitrogen. So because of that, we can add or replenish the nitrogen content of our pile by adding more leaf. So here the next one will be the list of reference. So I think that's all for our presentation. Thank you.